Hello? Aha, here we go. Okay, well, we might push off, so thank you everyone for coming along today, and thank you to everybody that's online. So I'd like to welcome you here today to today's seminar being held as the second program update of an exciting BHP-Curtin University initiative centring on the use of environmental DNA, or eDNA, and its use as a biomonitoring tool. So I'm Shane Herbert, I'm the program manager for the initiative that the team is going to be talking about today. So before we get started, I just need to do a little bit of housekeeping. Oops, sorry, before I do that, I just would like to acknowledge the Whadjuk Noongar people, the traditional custodians of the land on which we gather today that hold so much tremendous biodiversity and natural wonder, and I pay my respects to their ancestors, elders past, present and emerging. So getting on to the housekeeping matters. So in the event of an emergency, please exit through the doors that you entered. You just need to go down the stairs. Don't go all the way to the bottom. It's a dead end, so one level. And then ultimately the muster point is away from all of the big buildings. So it's actually across the bridge, across Mounts Bay Road, and on the grassed area that's next to the bus station there. Bit of a walk, but theoretically that's out of danger. Also, yeah, so don't take the elevators if there's a fire. So toilets are located through a door in the hallway off to my left. Whoops, thanks Zoe. We've organised refreshments at the conclusion of the presentations at W. Churchill's, which is kind of downstairs and across, different place to last year for those of you who were here. But in the meantime, there's still and sparkling water up the back with some cups. There's also a kitchen just off to the hallway, which you can help yourself. There's taps there where you can get some drinks. And it's please help yourself using the glasses there. This venue's got a 6 green star rating, so we've chosen to follow suit. However, we hope you can join us for drinks and nibbles at Churchill's. So we'll have the team guiding you to that, but basically downstairs, and it's down underneath us here. So all of that is far less relevant to our people that have joined us by WebEx, and welcome to you all. We've muted the lines, but if you have any questions for the speakers, if you could please put them in the chat, and my colleague Cheryl here will ask it of the speaker. Or if you want to speak, then request that and we'll organise that. We also aim to capture today's proceedings in a recording and make it available afterwards. So if anyone has any privacy concerns, then please advise myself or a member of the Curtin team. Okay, thank you for your understanding, and I think now with all that said, we'll begin the presentations. So to begin, I'd just like to provide a brief background to eDGES, as today might be the first time that you've heard about the program. Curtin University's Trace and Environmental DNA Lab, known as TREND, has had a strong expertise in environmental and ancient DNA. It's been applying it to environmental fields since it emerged over a decade ago. Consequently, back in 2019, a number of ideas for the group were assessed by BHP's Social Investment Framework, which I'll ask Dr Tim Cooper to say a few words about in a moment. Consequently, the program commenced mid-2020, so we're into our third year now, just into our third year of the original planned 5 years. So the program's had a dual approach to develop analysis mechanisms around eDNA techniques, detection techniques, with a direct line to addressing biodiversity change and mechanisms for the sustainability of key species and organism groups. The second was to translate this knowledge into outcomes that can ultimately benefit those that seek to monitor or manage ecosystems. The program's got 5 distinct projects within it. Each builds and expands on eDNA's utility as a biological monitoring tool in different ways, which we're giving you a little bit of insight on today. The driver of the research of the program is research, however, so ensuring that the findings lead to high-impact science get distributed to the community through peer-reviewed journal articles are important metrics for us. We also seek to use it as a way to facilitate connection to the eDNA network and the community, of which today is a major part of that, so I thank you all for coming along. So today's attendees is a broad cross-section of researchers, government, regulators and industry. And this is really pleasing to see everyone here today. Finally, as the researchers in the projects begin to as the research topics begin to mature, then the aspiration is to assess how we can contribute to biomonitoring policy and help the findings of this program endure well beyond what we're doing now. So the eDGES projects themselves cover 4 different environments, and each has a unique angle of extending or augmenting DNA methodology. So the first talk straight after myself, or after I introduce Tim, will be Professor Morten Allentoft talking about Pilbara Olive Python detection and conservation. That's followed by Dr Nicole White, who will be talking about subterranean fauna detection and conservation. Then we've got Dr Mattia Sacco, who's a prolific publisher, functional ecology of Chilean precordillera lakes. And then to finish, we've got Professor Euan Harvey, who'll be talking about 2 projects, improving sensitivity and specificity of eDNA tools for invasive detection and the Benthic communities associated with infrastructure in the Pilbara. And then we'll have a few closing remarks, asked for questions, and after each of these presentations we'll also have a few questions, so both after the presentation at the end, and then please join us downstairs for networking. So at this point I'd like to introduce Dr Tim Cooper to the stand, who's a Principal of Biodiversity and Land Management at BHP, to say a few words. Thanks, Tim.
Yeah, thanks very much, Shane. It's great to be back here again, year 2, to welcome you all to the annual symposium where we're talking about our collaboration with Curtin University on eDNA for Global Environmental Studies, or the eDGES project as we call it. I'd also like to acknowledge the Whadjuk people of the Noongar Nation, the traditional owners of the land and waters on which we're meeting today, and pay my respects to their elders, past, present and emerging, and I extend that respect to the traditional custodians of the lands on which other people are meeting, and in particular to those people in northern Chile where some of this project is located. So as Shane mentioned, this project's funded through our social investment framework, and that's our voluntary contribution towards projects with the primary purpose of contributing to the resilience of the environment and communities of which we are part, and we do that through local, regional, national, and in this case, global programs. So for the theme relevant to the eDGES project, our Future of the Environment pillar, the outcomes of our social investment projects are assessed against measures that focus on enhancing knowledge, improved management and protection or restoration of important biodiversity and ecosystems. And as Shane alluded to earlier, this project ticks a lot of those boxes, and in relation to enhancing knowledge, it's hard to find a more productive research team. And at the end of year 2, there have been 6 papers that have been published. One's submitted, awaiting acceptance. There's a further 2 very close to submission. And there's been numerous speaking engagements at national conferences, and a lot of that is due to the people sitting in front of me. So yeah, well done. The team's also made it onto the local news when they were taking blood samples of olive pythons at the Perth Zoo. And of course, this is our second forum. So we're getting some great outreach with this project. And I need to, next slide. Yes, there. So I've got a slide here of the Mountain Springs, a waterhole in South Australia, as a reminder to call out the great work that are being done by one of our passionate enviros in BHP. I can't see who's on the line, but I hope, Kim, that you're dialed in and that you're here somewhere. Maybe you can put something in the chat to let me know you're there. And she's been working with Nicole White on collecting water samples from these springs as part of Project 2, which is looking at the diversity of stygofauna in the southern part of the Great Artesian Basin. And these springs are the interfaces between the groundwater and surface habitats. And preliminary eDNA analysis is shedding some very interesting light on the evolutionary processes associated with these environments. So for example, analysis of 25 springs and 14 aquifers in the GAB show that there are 53 taxa that are unique to the springs, 6 that are unique to the aquifer, and then 15 metazoan taxa that are common between those 2 sources. Another interesting finding is that 15 of the 25 springs are distinct biological communities, and with 4 of the springs showing a very high proportion of genetic uniqueness when compared to other samples in the sample group. So this is raising some really interesting evolutionary questions, and we can't wait to see the papers that come out of this particular part of the project. And we look forward to the sampling as it progresses into the Pilbara region in Western Australia. So in summary, this is one of our truly global projects, and we're super excited to partner with Curtin on this project, and we look forward to hearing more about the progress that you've had in the last year. And so with that, I'll hand over to Morten. Thanks, Morten.
Thanks, Tim. Okay. Thank you so much for joining. I must say it's really a pleasure to be here in a real room with real people sitting in front of me. I had a long bumpy ride coming to Australia. I was hired at Curtin more than 2 years ago, but then something called COVID hit the world and closed the borders, and I've been sitting in a couch in Denmark for a very long time trying to lead the Trend Lab. But now I'm finally here, and it's, I look very much forward to this event here. So in Trend Lab, we are renowned for working with this environmental DNA substrate that is kind of the common theme across these 5 different sub-projects. That is, as I mentioned, the common theme in the eDGES program, really. And this is meant to be an overview slide that sort of shows all the applications, or some of the applications at least, where eDNA has a role to play. And what is environmental DNA? It's DNA in the environment. It kind of says itself, right? And I guess many of you are already familiar with this, with these molecules, but they are essentially, you know, all living organisms of this planet would have DNA in their cells. Whenever they decompose, whenever they urinate, defecate, shed their fur, if they are mammals, whatever, they will deposit DNA into the environment. Because DNA is the substrate of evolution. All the organisms would have different unique genetic sequences, genetic codes. We can, if we have reference databases available, with these sequences available, we can grab that DNA from the environment, extract it, sequence it, and match it up against those reference databases, and hence describe the biodiversity that we find in any given environmental sample. There are many caveats with this. It can be difficult to extract these short degraded molecules from the substrate. It depends a lot on how much information you have in your reference databases, whether you can actually match it up against the databases there and identify the species that should be present, and a number of other caveats. But we have a very well functional lab and some very regular pipelines set up to do this. And this is just to show, right, where can we find environmental DNA, essentially in any biological or environmental substrate that you can think of, whether this is terrestrial environments, marine this doesn't really work, huh? Marine environments or freshwater environments. It could be in caves, subterranean systems, or whatever. Anywhere you're interested in monitoring biodiversity, essentially. And in that sense, eDNA becomes a supplementary tool to traditional biomonitoring, which can be time-consuming. You need a person in the field, observing. It needs to be someone rigorously trained in identifying the right species. So you need a lot of taxonomic knowledge, depending obviously on what groups of organisms you're looking at. Some of this, traditional taxonomists that know what they're doing in the field are hard to train, and they're becoming more and more rare these years. I mean, so in that sense, you can say eDNA really has a role to play. Not necessarily replacing traditional biomonitoring, but running as a supplement. And there are ways to balance things, with the different methods. So this is the eDGES team. I'm not going to stand here and introduce every one of them, because you will meet most of them as they speak, and you will meet all of us after. But let me just reiterate again what Tim and Shane has already said, that it really is a wonderful experience working together as a group in the eDGES team. We all really enjoy gathering up and sharing ideas and new results, and it really is a great project whole program that we have going here with the 5 different sub-projects, and it's a pleasure to stand here today and share some of these results we have so far. I will be talking about the Pilbara Olive Python. Sub-project number 1 is called eDNA Monitoring and Conservation Genomics of the Pilbara Olive Python, I guess many of you would have heard or even met this snake here. It is one of the world's largest snake species, probably the second largest in Australia, next to the amethystine python. It grows up to 6.5 meters. It's a subspecies, whatever that means, of the common olive python, or the Kimberley olive python, which has a more northerly distribution, as you can see on the map down here. and it is presumably an endangered terrestrial top predator. I mean, as you know, all over the world, top predators are in trouble, right? Because they're in top of the food chain. If anything goes wrong in one of the lower parts of that chain, the top predator is certainly hit. And the same goes for the olive python here. Even more so, they might also be threatened by changes in the environment, such as potentially mining activity or other anthropogenic impacts that takes place in the Pilbara. And this is one of the reasons why we really need more information on this species, ecological information, in order to distribution information, in order to properly assess the conservation status. And again, pythons are difficult to monitor. Snakes in general are, in many cases, very difficult to monitor. They're difficult to find. You can spend weeks at a time in the field and not finding very many individuals. This one is actually an olive python here I found up in Kakadu National Park some years back, and I had spent about 3 days looking for pythons. This one, then while I was standing cooking in the middle of the night in our campground, crawled in between my feet, and that's how I caught this one here. So it just shows there's a lot of luck involved and a lot of randomness involved when you're trying to find these animals. And this is, of course, a good example of where we think eDNA could have a real role to play when it comes to monitoring for these elusive and difficult to find species. So what are the aims of this project? We want to first and foremost advance the ability to detect these pythons in the environment with eDNA technology. We high precision the eDNA tools that can detect the python DNA. We want to sequence as part of this project the first complete genome of the Pilbara olive python. I'll get back to that towards the end of the talk, why we want to do that. And this goes together with the third point here. When we have the complete reference genome, we believe that we can do a large-scale conservation genomic study of the olive python populations that can supplement the eDNA component of this project in various ways. And I'll get back to that. And then the last point, which we haven't really touched on yet, but which will be unfolded a bit later in the project, is we will develop and deliver a community engagement program as part of the work. Most likely, particularly as part of the field work. Okay. First question. We know from all kinds of years of studies, not my own, but what have happened in Trent before my time and in other eDNA projects, that reptiles are fairly difficult target animals when it comes to eDNA technology. And one of the reasons is that they seem to not be shedding a lot of DNA into the environment. You know, reptiles are highly adapted to avoid evaporative loss. They have to sit in the sun to heat. So animals like that, they have to preserve water extremely well, which also means that not a lot of excretion are happening into the environment. And this is probably one of the reasons why it seems to be very challenging to identify reptile DNA in the environment. So you can say, isn't it a bit stupid to frame a whole project around an animal that we know already from the beginning is going to be super challenging? And in that case, we say, okay, probably the best place to start then to test these things is to go for rock pools. These snakes are, I wouldn't really say semi-aquatic, but they tend to congregate around the rock pools, mainly because that's where they feed, where the food is. So they ambush predators. They would lie in the water for hours and wait maybe for a small wallaby to come down and drink and then attack them. And this implies that we probably think that we'll have a higher chance of detecting environmental DNA in these rock pools rather than going out and run random area in the Pilbara and taking a sediment sample and taking it home. This seems to be a good starting point for this whole project. And luckily, eDNA Frontiers, our sister company, you can say, our commercial arm of Trent Lab, had already been working with the various consultancies for testing these, for biomonitoring purposes of identifying pythons. So we could essentially, we were allowed to take those data and present them here today and show you what we got so far. And we're using this metabarcoding assay, this set of primers. Primers are the ones that bind to the DNA and identify the region in the DNA that you are then amplifying and analyzing in downstream analysis. So we're using a method for that that was published a couple of years ago in Trend Lab. But the lab work for what I'm showing you now was done with Shane and his team in eDNA Frontiers. And we have here data from 6 sites, rock pools in the Pilbara, using this particular metabarcoding assay here, and using the eDNA bioinformatics workflow. This is what, after we've generated the sequence data, we need to analyze and see what species we actually have, what taxonomy we have present in our data set. And here we are lucky to have Martha with us. She's in the room over here because she's part of the eDGES team, and she has developed this pipeline here for analyzing metabarcoding data efficiently and identifying the taxonomic composition. And this is what we get from this little pilot study, you can say, where we compiled previously generated data in the Frontiers lab. And I don't want to go through all of this. You can see we have the 6 site names up here, and you can see the taxonomic composition defined by the different colors. You have a lot of different taxa down here. I'm not going to go through everything. And it gives you an idea of the biodiversity in these particular different rock pools that have been picked up by this particular primer set that we're testing here. You could say it's a little bit funny that we are using a reptile primer set, and then we're getting birds and cow and dog and whatnot. Nematodes are all kinds of things, right? But it is because primers are not perfect. They will bind to occasionally bind to any kind of DNA that is present. In particular, if the target DNA is not present, then they start binding to other kind of DNA, implying that you will amplify other things in your PCR reaction. So you get a broader composition than what you actually expected. In particular, if the primers have been perfectly optimized and tested in every different way as all kind of other sweep of taxa, then you might have something that only binds to pythons. But in this case, it's a more broader primer set that should pick up reptiles, and we can see it picks up a lot of other stuff as well. Anyway, the important bit here is that we're looking for the yellow color, because the yellow color is lyases. That's our olive python. And you can see everywhere at these sites where we have a yellow color, it implies that we have python DNA present. So you can see you find it in the Newman rock pool, this one simply called Pilbara here at the Western Ridge. And so 3 of the 6 localities, we can certainly identify olive python DNA sequences in the data. Again, this is relative composition, so you can see in some of them, in this one site here, it's essentially only python DNA being present, whereas some of the other sites, it's a composition of a lot of different things that we are observing. We don't want to overinterpret this. This was done as part of consultancy work. It's not necessarily suited for a big-scale study looking at distribution in time and space and everything. It doesn't really matter for the sake of this little, in this particular context. We just wanted to show here, yes, there is python DNA in the environment. Yes, we can pick it up and identify it. This is a very good starting point for any type of studies we want to do here. So this was a bit we could wipe the sweat off our forehead and tell Tim, okay, you have funded this project. We are glad to tell you that at least we have some place to start here, right? We have something to work with. The next step we wanted to go with this is that we want to try and look into, and this is still a work in progress, but we have taken the first step of this to actually look at how long is this environmental python DNA preserved in the environment. Why is this important? This is a bit important because if you're using it as a biomonitoring tool, it's fairly useful to know if you're monitoring something that was there 3 years ago and the DNA is simply lingering around, or it was 3 days ago. And you need some sort of temporal precision, ideally, as to what you're actually looking at.
Some people online having a bit of trouble hearing you, Morten.
All right. Should I try this instead?
Hello?
Is this better? Online crowd. All right. Okay, I'll start over then.
We're hearing you. It's just really warm, so I think the microphone.
All right. Okay, dokes. We had Ben helping us out with this. Ben is a recent or still honor student in our lab, in the Trend lab at Curtin. Ben came to us because he really wanted to do some computer analysis. He was mainly interested in doing bioinformatics. So after 2 days, we took him away from the computer and put a pipette in his hand and said, this is what we do in Trend lab. Computer work is later. I've never seen a guy shake this much with a pipette. He had 3 supervisors looking at him. So it was a few tough days for him in the beginning, but as I will show you in a second, he actually produced some really high quality data and did really, really well. I mean, he's produced a very nice thesis. We haven't had the evaluation from examiners yet, but I would say that it was a nice piece of work, and I'm showing you some of the results from this now. So what we did, the original plan was to take living pythons, and we still hope to be able to do that a little bit downstream, chuck them into some water tanks to mimic what goes on in a rock pool, and then sample these water tanks after various points in time to see how much DNA was preserved in these water. This turned out to be a little bit of an ethical nightmare. It was very early on recognized as an animal experiment by the ethical committee at Curtin University, and that's all fair and good, but we didn't have time to disentangle all this paperwork before the project, for this honors project. So we did something else instead. We said, can we find another proxy for DNA, Python DNA in water? And what we did instead was we took blood, because we had some blood standing around our freezer that we were going to use for the genome work anyway, which we'll talk about in a second. So we took this blood. We know it's probably not the best mimic of what goes on. I mean, not many pythons bleed out into the environment, right? But it doesn't really matter for the sake of this proof of concept. We know we have high quality DNA to start with. It's a good starting point for evaluating the kinetics of DNA decay that are happening downstream. So what we did here, we took 2 20-liter tanks, one with completely pure water, sterile water essentially, and one with lake water that we took from some suburban lake around Perth. I don't remember which one right now. It doesn't really matter. But to kind of mimic natural decay processes that would happen in a real lake or rock pool, or and then a setup here where we don't expect DNA decay to happen very fast due to biological processes. DNA decays spontaneously. It's not a stable molecule, but our hypothesis was that it was going to happen much faster in this in the natural environment than in a sterile environment. And this is what and then we did qPCR as the means to attest to estimate the concentration of DNA as we went along. And this was an experiment running for a month. I'm going to show you some of these nice decay curves here. This is the pure water tank. You have DNA copies on the y-axis and the time on the x-axis here. And you see this very nice decay curve here. DNA degrades over time. It degrades with a constant rate, which is why you get an exponential relationship like this. You can essentially say DNA has a half-life. This is well known already. This is not really new knowledge. This is how all biological macromolecules decay. Every single bond in the DNA strand breaks down with a certain probability at every given time unit, and this is why you're dealing with rate kinetics like this. But it was very nice for us to confirm that we could get this information in our data. It shows the primers behave properly. It shows that our whole experimental setup behaved properly. It shows that Ben created some good data despite being a bit shaky. And we have 2 curves here. One of them is the total data set. You can see it's a bit wobbly in the beginning. It seems to be jump up and down through time. There's no really pattern before we're actually getting the nice exponential decay. And it's because the blood clotted. The blood clotted in the pure water tanks, which means that there was a very non-random sampling in the beginning. So you have one that exemplifies our total data set, one that takes this blood clotting into account and only really start the analytical fitting of this model a little bit later in the process. And this is also why we have a much better R-squared value on this when we take that into account. Okay, so what happens when we go to the lake water? Same deal. We have an exponential decay pattern. We're very happy to see this. and a very nice fit as well here. They look very much alike, except when you look at the x-axis, right? If you start looking at the time scale here, they're very, very different. And this becomes even more apparent when we plot them together, because here we have the pure water tank, and here we have the lake water tank. And it's very obvious that when we're dealing in mimicking natural environments here, for whatever reason, we get a much, much faster DNA decay of this Python DNA here. I mean, we're losing DNA molecules at an incredible pace, have a very short half-life, this particular DNA fragment. So, yeah, as an example here in the lake water tank, we lose 99% of the molecules after 2 days. What does this mean? Of course, again, we have to remember the caveats here. This is blood. It's not a real rock pool we have sampled and all these things, but it certainly suggests that when you have a natural environment, the DNA only stays, the environmental DNA only stays around there for a couple of days, and then it's gone, implying that the animal you're analyzing and you're seeing here probably visited this rock pool fairly recently. Again, numerous follow-up experiments needs to be done here, but it's probably microbial decay that are happening at a fast rate here in this natural tank. We know that primers pick up other species. We've seen that already from the eDNA frontiers data. So how can we know that we're actually monitoring Python here and not something else? Here Ben did some other experiments based on what we call DNA melt curves, and DNA melt curves is a way all DNA melts, double-stranded melts into single-stranded DNA at certain temperatures, depending on the fragment length, depending on the base pair composition. And here, this is a pure water tank. We see at time 0, we don't have any signal down here, or at time -1, I should say. This is before we add any DNA into the water tank. There's no peak. At time 0, when we start adding the blood, that's when we get a nice melt curve signal here through time. This is back through time all the way up to a month, essentially here, right? So this is the Python DNA, giving us a nice peak, and that is present in the pure water tank all the way until the end of the month, essentially. Then when we look at the lake water, we see the tank control. We thought initially, shouldn't that be empty? Why are we getting a peak over here? But this is, of course, everything else that these primers are picking up. We know there are turtles in there. We saw from the eDNA frontiers data, these primers are picking up all kinds of other things, right? So we are actually amplifying something here at the time -1. Then at time 0, when we're at the Python DNA, the melt curve shifts because now we're actually observing proper Python DNA here through time at various time intervals. And then at some point, the Python DNA starts disappearing again, roughly 24 hours. So the primers again starts competing with the other DNA that is present, and then the melt curve shifts again over here, right? So it's essentially this competing kinetics between the DNA that is available in there and the Python blood and whatever else is preserved in these water samples. So there's a nice fit with these things. And this is again exemplified in sequencing data. I'm not going to stand here and talk a whole lot about that, but we were a little bit interested in seeing if we could identify the same kind of signal when we actually sequenced the data and got the sequence information. qPCR is just looking at curves and melt curves and CT values and these things, but you never see the sequences as such. So you're always a little bit worried what you're actually analyzing. In this case, we sequenced them, we got the data, we could reference them to our databases and identify the different species in here. And you're seeing here abundance in terms of number of sequences in the pure water tank. After we added the Python DNA, we're only getting Python DNA. That's a very nice confirmation. If we started getting a whole pile of other things, we would shut down the labs. And then in the lake control samples, before we had Python DNA, there's no Python DNA. Then we start adding the Python DNA, and you see we're essentially almost only getting Python DNA up until a certain point in time, when other things starts turning up, right? So it behaves, again, in the same way as we expected when we look at the decay curves and when we look at the melt curves and all these things. It all goes together to kind of show 3 lines of evidence that this DNA decays very, very rapidly in a natural environment. Many further steps needs to be taken to analyze this properly. Blood, as I mentioned, blood is probably not the most suitable substrate to use, but this is what we had available at the time. And you could play around with different pH value, different types of water and whatnot, right? To really get a feel for the real kinetics here. But opens the doors to a lot of new interesting questions and show that the DNA of this python in a natural environment are disappearing very fast, right? Okay. In summary, we're happy we can successfully monitor olive python using eDNA in water sources. That's a good starting point for everything we want to do downstream. We know python DNA degrades very fast in natural environments, and as I mentioned, these are preliminary results, right? Lots of work ahead. And what is that work? One of the things we really want to try to explore with this project is something we call eGenomics instead of eDNA. And this is kind of the motivation for going to the Perth Zoo and drawing blood from this beautiful lady here. I think she was called Bobby, right, Martha? Yeah, Bobby. And which is a big, beautiful female olive python that they have in the Perth Zoo. We got some blood sample because that gives you super high quality DNA also. We took it straight into the -80 freezer. And then we sent a sample of that to the Vertebrate Genome Consortium in the US, who specialize in assembling de novo genomes. This is a difficult exercise. You need to be able to sequence very long strands of DNA and have various pipelines set up in order to kind of assemble the puzzle that it really is to assemble a de novo genome for the first time. So why do we want to do that? I'll get back to that, by the way. But right now, when we're only about to get access to this genome data, we have a very high quality genome. It has an N50 value. It's 1.5 million base pairs. This is expected. This is known for other snake species that there's roughly the genome size in snakes. So that's a good confirmation that it's probably a solid data set. And then we have what's called an N50 value of 72 megabases. It's an indication of a very high quality, very well assembled genome, meaning that 50% of the genome assembled is 72 megabases or higher. So it's very long strands of DNA that have been assembled here in a good quality. So the puzzle, you can say, of assembling this genome has been done extremely well. And why do we want this? Why do we want the full genome in this project? What does it give us when it comes to environmental DNA and these other things we're discussing in the context of this program? Essentially 2 reasons. The whole eDNA field is very fixed on these barcodes, 200 base pairs or something, because that are species diagnostic. This is what we can detect. This is what the primers pick up, and the databases and the whole environment is set up essentially to deal with these specific barcodes. But animals or organisms don't release barcodes into the environment. They release the whole genome. So by doing it that way, we are potentially missing out on a hell of a lot of information that is available from full genomic data. A lot of people, most eDNA researchers don't want to go down that avenue because it's risky, it's pricey, and there's a very big chance that we cannot identify all the full genomic information, and the sequence data drowns in bacteria and whatnot that is in the environment, because you have to sequence it a different way when you're looking for genome-wide information. But if you don't test this, if you don't explore it properly, then we're never going to get anywhere. Then 10 years downstream, we'll still be looking at barcode data and not trying to push the limits anyway. And the first step, if you want to do this, is to have a whole genome. Because if you don't have the genome, you'll never be able to identify all the genomic-wide information that is floating around in the environment. So that's the idea with this. Let's try to get a Python genome. Let's try to apply a different sequencing strategy called shotgun sequencing, where we take some random water samples, environmental samples, do some testing in our lab as well in different lab settings, and see how much genome-wide Python DNA that we can actually retrieve under a number of different scenarios. And that's one of the avenues we'll be exploring in this project moving forward. The other reason is we wanted to get into some conservation genomic analysis. We want to look at estimating population sizes in the Kimberley versus the Pilbara. We want to look at adaptations. How are the different subspecies actually adapted genomically to living in different environments? When we talk about conservation genetics, we have to remember a little bit what are we actually preserving. We're not you can say the body itself is just a holster for the DNA, potentially, right? I mean, that's essentially how it is. We're preserving the blueprint for the future. I mean, it's a DNA that we are preserving. I mean, when you look at the body of any given individuals, it's just a snapshot of the evolutionary phenotype at this particular point in time. But in reality, it's the DNA we are preserving, and we would really like with this project to bring that a little bit into the whole conservation context. When we're talking about the Kimberley subspecies, the Pilbara subspecies, what are we actually preserving? What genes are adapted differently due to natural selection processes, for example, in the two different environments that we want to preserve for the future? You can say this is very academic and not really applied, but we are so lucky that we have that freedom in the framework of this program to not only go down the applied route, but to also really explore these basic research questions that we think are very important. So stay tuned, much more to come in the years ahead. Thank you so much. Yeah.
Thanks, Morten. Did anyone have any questions? We could have questions now, or we could have questions later.
Great talk, Morten. Very interesting. The DNA degradation study, did you control for UV radiation in that study, or did you get any indication of just, whether that was only a very small influence on degradation compared to the microbial breakdown?
It's not something we have assessed yet, but it's definitely one of the parameters we would like to investigate more. Right? At this stage, I can only say the two tanks were kept in the same room, same temperature, same UV radiation, and all these things. So the differences we see here are certainly not due to UV. But UV is a big killer of DNA. So we imagine that in a rock pool, actually, that sits in the sun, I mean, that will definitely have a role to play also. Yeah.
Any other questions? Cool. All right. Well,
thank you.
We'll open it up for questions again at the end, but I'll get Morten to hand over to Nicole now.
Yeah. So next up is Nicole White, who will talk about subterranean stuff.
Yes, subterranean. Good afternoon, everyone. Technical challenge, trying to get a microphone on. I'm Nicole. I'm here to talk to you and share with you a bit of results from the eDGES Subterranean project. And just before I kind of jump into some of the work that I've been doing, I would like to acknowledge the team that's been involved in this project. I can't take all the credit. I have some awesome collaborations going on with people in South Australia, Kim Solly, who's from Olympic Dam, as well as Michelle Guzik. Sorry, the Adelaide Uni logo's covered. From Adelaide Uni, and Nick Murphy, who's at La Trobe University. Between Michelle and Nick, they've got about over 30 years experience working in the Great Artesian Basin, particularly with molecular work. I also would like to acknowledge the traditional owners of the land. I'll be sharing some results with you from samples from the Gabs Ring, so I would like to acknowledge the Arabana people and those traditional lands. And then also we have our research team at Trent, so a big shout out to all of you. So the people that have been involved, we have our little subterranean and research group at Trent, which involves Mattia, Mika, myself, and Julia, who I'll be introducing. So there was two main aims of this project when the project first got written up and presented and put forward, and this was a smaller project. It wasn't a full-time project for me. So we had a couple of aims to do, and really it was, we've been hearing about databases now already. If you're not familiar with it, we rely a lot on reference databases so that we can assign the environmental DNA that we isolate and amplify to a species or a taxon group. Otherwise, we need something to reference to. And the subterranean world and references for subterranean taxa certainly is an area that needs to be addressed and improved. And then with mitochondrial genome, so Morten's gave you a little introduction to genomics. This is kind of where we're going in the subterranean world too. We're producing larger regions, the whole mitochondrial genome via shotgun sequencing, rather than the short or standard 700 base pair barcodes. There's a couple of advantages for that, and then there's also a couple of challenges, so I'll go through those with you as we move through. So this project involves South Australia, the Great Artesian Basin. It's the southwest corner of the Great Artesian Basin, and I do hope to address Ethel Gorge up in the Pilbara. That's an aim for next year. So there's 2 locations for this project. So we'll just focus in now on the springs. So there's a lot of springs in the Great Artesian Basin, lots. And it's really interesting. They have some pretty interesting names, and some of the names of these kind of clusters or complexes of springs may contain only a few springs. Sometimes they can contain quite many. So we started looking into, well, how are these springs or spring clusters, complexes classified? So loosely, a cluster. So, for example, Lake Eyre South, this is a complex, this is a complex, this is a complex, and this is a complex. Their water chemistry for it to group into a complex, and they source the water from the same fault, and that's how they get grouped. Okay, so we had spring water from 4 complexes. So I'm only going to be presenting some data from Coward, Lake Eyre, Hermit Hill, and Wagiana. As I said, the Gabs, it's a very large area, as we can see from the map up there, and the Gab extends multiple states. Yes, a very, very large basin. Okay, so the samples for the water, and we've also got groundwater. I'll introduce you to the groundwater shortly, but COVID, unfortunately, restricted any travel outside of WA. So I was fortunate enough that the field teams over at the Olympic Dam routinely monitor, and I was able to get samples from the fields through Kim Solly and her field team. So that was actually fantastic. So even though I couldn't get out, I was still able to get the samples, so that was great. So springs, they can fluctuate a little bit. I don't know what the natural seasonality is with the water, but I was reading some literature, and it had said that some of the complexes, spring complexes, show a 6-monthly lag to rainfall, regional rainfalls, which I thought was really quite interesting. So even though we did get quite a bit of water samples that we wanted, there were 29 springs that were visited during those months that there was no standing water, or there wasn't enough water so that we could get 5 replicates from, and that's what we need. We need some replication in the sampling, so we weren't able to do that. Now, what is it about this rainfall in these recharge areas which makes it really interesting? And I didn't know this, but the Gab Basin kind of recharges from the basins and the rain. So you can see if the rain, and our study area is down here, and the recharge areas are on the outside, it can take quite some time for the water to go in. I was also really, really interested. Tim had a great photo at the beginning of what you see on the surface, but it's really interesting on how these springs and the geology and it works. So as you can see here, The actual water source can be many, many hundreds of meters down and below rock. And then it's kind of like a weakening or a bit of a fissure, and then water comes up and generates. And we saw in that photo that Tim had at the beginning, you actually saw some bubbling in the spring, and then that's what can happen. And then they kind of overflow, and then you can get the swampy areas around the spring, but it's kind of the water flow comes up and we have these springs, but they can be extremely deep, the source, the water source. So not only did we get water from springs, but there's actually 2 bore fields or well fields within the vicinity of the springs. Again, we've got samples from Coward, Lake Eyre, Hermit Hill, and Wagiana. And we kind of have this bore field A, and then we have bore field B. So Kim had asked, she said, oh, you want some deep groundwater? And I said, wow, yeah, that would be great. That would be great to get some of those samples. She said, all right, well, we routinely monitor, and they're pressurized bores. I said, pressurized bores? We're used to dropping things down a hole and having to pull up samples or water samples from the Pilbara. What do you mean pressurized bores that they're shut in? But this is actually what happens. So they're capped bores because they're under pressure. So we were able to get groundwater samples that went down to 312 meters, which I thought was quite good. When I was doing a little bit more reading and looking into these waters in the Gab Spring, there's been some dating studies that show that some of the water is up to 2 million years old, which is very interesting. And there's lots of pressures on the groundwater because there's a local township that they pull on the bore fields. There's pastoral needs in the area. They pull from the bore fields, and then we have the industry operations that are also going on in that area. So there's multiple pressures on the pull on the groundwater there. Another, you can't quite see it under this, but bore field B, the groundwater down there is 60 to 80 degrees, which is actually quite warm. And in some areas, other areas of the Gab, water can be up to 100 degrees. Okay, so you got to be a little bit special to be able to survive in 100 degrees. Yeah. All right, so this basically shows you the difference between, so the light green dots are the spring water, and then where the bores are, and kind of the distance and the spread across. So what did we do? In the end, we ended up with 125 samples that came from 25 springs where I had replicates. Then I ended up with 70 samples of the SIP bore samples, so those deep groundwater samples. And I have COVID contingency plan written on there because running a metabarcoding assay and the 18S assay, as you saw in the main 2 aims on the front slide, wasn't actually part of this project. But because of COVID and delays in other things, the order of things had to be redirected, and to keep things going with this project, we got the water first, which I wanted to get the animals first so I could do the mitochondrial genomes, develop the markers, and then run it across the water. But it didn't quite work out that way. I've got the water first, so what we wanted to start exploring is kind of the differences between the spring and the groundwater, and then the springs themselves, but also how sensitive is and how much DNA, environmental DNA is in these waters, given there's these temperature differences, and see what we could find. So that's what we did. So I ran the 18S metabarcoding assay across all of the water samples. Initially it was, okay, well, let's have a look at the groundwater and the spring water. And this kind of really, so this is what we do. when we start looking at metabarcoding data, and we refer to them in ZOTUs, operational taxonomic units. So when we looked at all of the data, we did blast it against the reference database, GeneBank. 40% of the data, we couldn't assign it to anything from what we had amplified from those samples. And then you can see the breakdown there of basically 16% got assigned out, and then 11% to, going down to the metazoans and then the fungi. And then what we decided to do is there was a lot of controls in this experiment. There was filtering controls, lab controls, extraction controls, PCR controls. We had a lot of controls in this, so we removed anything that was positively amplified in any of the controls along the way from the data set. And we also removed the samples which we consider a low sequencing depth, so anything under 15,000 DNA sequences per sample was removed and considered as a low depth sequencing of sample. So what did that leave us with? That left us with 149 samples that kind of passed through all of the filtering. Our quality controls we're pretty happy with. That resulted in over 7,000 operational taxonomic units. And then if we were just interested in looking at the metazoans, so that's our kind of our multicellular animals, we ended up with 831 ZOTUs. So 18S, we've been talking about, as Morten already introduced you to, the reptile marker that was used for the honors project, and it picked up other things. 18S is a very broad assay. So it really depends on the question that you're asking and what assay you apply to the samples. So we know this, 18S is a very broad assay. Okay, so it's gonna pick us everything, but if we want an idea of the community that's there, it's a very good assay to use. You can do this kind of first pass, use a broad assay. If things are kind of coming up that look interesting, then you can select another marker set and go back to the extract and reinvestigate with a different marker set if you want to. So it really depends on what it is. So take-home message here, there's not much in that groundwater. Springs, yes. That deep water? No, not from the samples that we received or the points in time that they were sampled did we detect a lot of environmental DNA. Yeah. So this is it again. Just looking in another way. So when you see all up there, that's everything, the complete 18S data set. And then we thought, all right, well, does it change when we just take it all the way down to the metazoans and we chuck everything else out? And the picture's still the same. Picture's still the same. And then we started looking at, well, what is it that's kind of shared between the two sources? And what's kind of unique then? So if you just look at the metazoans, this is it here, 53 are kind of unique to spring water. There's a few shared, and then there's some things that are unique just to the groundwater. So this figure here on this side kind of gives us an idea. This is a cross-section across the Gab Spring here, and what it actually looks at. Remember I had said the recharge areas are on the outside. So water comes from the outside and it comes into the Gab Springs. So you have subartesian and artesian. The SIP bores are kind of what the bores are under pressure, so they have to be capped. Yep. So that was artesian water, and then we have the spring water there. So, okay, well, right. Not much in the groundwater, but there certainly is in the spring water. Why don't we have a look at this a little bit more? So I was quite interested. I kind of see these springs as little oases, and I was really interested. So I'm going, how unique are these springs in terms of the ZOTUs that they contain. And remember, this is just the 18S assay that we used. Over on this side, you can see here, this is how unique proportion of the total ZOTUs unique to that spring. All of them kind of have a level of uniqueness at a varying degree, but you can see these ones that I've highlighted in red, they've really jumped out, which we thought was quite interesting as to, right, they have a high proportion of ZOTUs that are unique to that spring when we compared the springs to each other. And remember, I can only compare what I've looked at and analyzed, and remember that was only from 4 spring complexes. But it really starts to kind of jump out at you. The 4 complexes, again, so it was, all right, we know there's some uniqueness now, some genetic uniqueness in the springs. But again, they're kind of classified here according to water chemistry and where they source their water. So can DNA or the DNA signatures from these springs also follow this classification? And so we did a cluster analysis on it, and what we're finding is we're having 3 based on the DNA, kind of 3 groupings. Right, so you can see in here kind of the coward and the CBCs are kind of falling in a little bit with the other spring complex. So there is some grouping going on, which kind of loosely reflects where they are in the landscape, but the DNA has grouped them a little bit differently when we base it on a clustering analysis. This is the same. Rather than a tree, we're kind of looking at the network analysis. We were kind of expecting to see 4 colors if there's 4 spring complexes, but we're seeing 3 colors, so there's 3 groupings going on here. Yes, so that was all of the data. So then we did the same thing again and go, all right, should we just examine the metazoan ZOTUs rather than the all and see what happens? And they're loosely, they're following each other again. So we're seeing this kind of clustering of 3 groups based on the 18S findings. So that's as far as we've gotten with the groundwater and the spring water. But there was also an invertebrate monitoring program that occurs once every 3 years over in the Gab. This allowed for me to, well, after they were identified, so they went to a consultant to taxonomically ID the animals, and then they were sent over. So really, the mitochondrial genomes is the aim of this project. So what I shared with you with the 18S and the water wasn't really the main aim, but we're finding out some really interesting things about the springs. I'm now underway with the animals from the Gap Springs. So I got 90 plus animals a while ago, and 80 of them have passed the gauntlet in that we have enough DNA, and I'm satisfied that they can go through the pipeline. Why I say that is I've been working on these little critters, and if you have a look at this, those are copepods, and that's a millimeter. Okay, I've got animals that come in, and I'm working on that, right? So high quality DNA and enough of it to do the shotgun sequencing and generate these libraries has kind of been a bit of a challenge. So I'm happy to report that 80 passed, and they're in the pipeline. But I've already been working on, and this is in collaboration, we have a few other subterranean projects that are going on that I've been involved with. So there's an ARC linkage grant, Michelle Guzik, who's in South Australia, and Mika Vanderhart. And by the way, Mika did the data analysis on the Gab Springs, so thank you very much, Mika. Mika's been busy in the field. She's been busy doing the metabar coding. I've been involved in this project, and I've been doing the mitochondrial genomes. Now, this really gave me a good warm-up for optimizing the technique in the lab. So we've got almost 60 now mitochondrial genomes and these all contribute to the reference database. Right, and mitochondrial genomes, rather than a 700 base pair barcode region, we've got 16,000 bases, and we've got all the genes on the mitochondria. So now we can start looking at, is the CO1 gene the gene we should be targeting for this group, taxon group? Or is it the 16S, or should we be looking at another gene on the mitochondria altogether? It's kind of just followed that this is the standard barcode region, but is it really? And then I could see that there's not many people doing mitogenomes on these animals, because this is really challenging. This is really, really hard to do, right? So this is underway. I'm also working on the gudgeon, which is a blind cave fish, subterranean fish. I've completed the mitochondrial genomes for that. So that's where taxon-specific markers will be developed. I'm not interested in the community or that broad brush analysis. I just want to know if gudgeon's in there. And then I'll also be working on developing assays that target a group of Stygofauna, the important groups that we're interested in. This was a little bit of work that Mika did, and this is just very quick. So there is a big barcode reference library database that's currently underway. Michelle Guzik is driving this one. Mika took this database, and these are our kind of common metabarcoding markers that we use. And then there was a paper that came out that said the EPD-TRN2 reverse primer is better than this one for picking up insects. So what we do, what Mika did, is you can run it through a software program, got all the reference sequences, and this gives you the likelihood with a score that the primer is actually going to bind if the DNA is actually there. So you can kind of start looking at this and know beforehand, if you're using this primer set, basically this forward primer, if this is present there, is going to have a hard time binding to the DNA and amplifying it. So this allows us to troubleshoot a little bit and see what groups of animals really need assay development as well. This is to just show you the importance of some of the animals over there in the springs. There are species of snail that are listed. They have a conservation status, endangered and vulnerable. I have both of these in the lab and multiples of, and they're in that group of 80 animals. So we will be doing the full mitochondrial genomes for those animals as well. Future plans, continue in the lab, get into the assay development, and get some field work going for the Pilbara. And that's me. Thank you very much.
Thanks, Nicole. Some really interesting work there. Has anyone got any questions for Nicole? When we were talking about those three groups, we were talking about the possibility of underground refugia. Did you want to expand on that?
Underground refugia.
Underground refugia for the Stygofauna.
Yeah, so we have been talking about it in amongst our groups and a lot with Michelle and Nick, and they are of the opinion that they're kind of refugia, like it was, the landscape was quite different. It's quite arid in the Gab Springs. And so when the water retracted or when the arid land started and then you had the spring mounds, it kind of maintained the biological diversity. So they are these kind of little oases little islands, so to speak, of refugia of animals. Yeah.
Yep. And finding out where they are could be
Yes,
tremendously helpful.
Identifying the uniqueness of them. Yes. Occasionally there are some flooding events that can occur and can carry some animals, and they've done some population genetics on the snails pre and then post a flood event, and could see some animals got moved around and they could see the gene flow. But I don't know how widespread that is. Yes.
Are those 3-4 springs that are the most unique related to their persistence at all? The springs, you mentioned some were dry, some were dry. Is that sort of...
Yeah, I haven't gotten into the details. I haven't yet. I mean, there's water. Yeah. But how close they were to the other, I don't know.
Cool. Okay, well, thank you very much, Nicole. We'll hand over to Mattia now.
Thank you. Good afternoon. My name is Mattia Sacco. There's an accent over there. No one pronounces it, but I don't care. Which can be a problem for my son because there's no accents in Australia, so let's see. Okay, so today I'm going to talk about the only international project part of this eDGES program. International because there's an international collaboration with the University of Antofagasta in Chile. This is a big project involving mostly hypersaline ecosystems. There's a bit of a debate on the definition of hypersaline ecosystems. We usually at Trend tend to go with the broader one, which is 35 grams per liter, which is more or less the salinity of oceans. Lakes beyond that salinity are considered hypersaline. Within this collaboration, is way beyond Nicole, Trend, and myself. There's Matt Campbell, research technician and researcher, more than technician at Trend, Mika, other collaborators. From the University of Antofagasta, there's Professor Chris Arrod. One PhD student is fully funded by this project, Gonzalo Salazar, and there's also a postdoctoral research fellow involved whose name is Pablo Aguilar. We have one big goal, quite challenging, but also quite exciting, I would say. We would like to combine eDNA, so we would like to use eDNA as a tool. eDNA is the pillar of this project, but there's other tools, analytical tools that we want to use. And we want to combine that with biogeochemical investigations, and you'll see in a few slides what I'm meaning by that. The broader goal is to characterize biodiversity and ecosystems functioning in hypersaline lakes of high conservation concern. You might be familiar with the Atacama Desert in Chile. Chile is a very skinny country, but very long. In the north of Chile, you have a very arid region where you can find quite big hypersaline lakes. Perhaps the most famous one, which is familiar to your ears, is the Salar de Atacama, which is top 3, I think, on top of my mind, top 3, the biggest. I think the first one is Lake Eyre, so top 3 biggest hypersaline lakes in the world. Quite a big goal in terms of ambitious goal. And that's the reason why we wanted to bring this into a level of multidisciplinarity that could take advantage of the different technological advancements that we have to date in order to go into functional ecology. I'm pretty sure that not many of you are not familiar with hypersaline lakes, even though in Western Australia we have a lot of them, if we think about the goldfish, for example. They're definitely not charismatic ecosystems, I would say. People tend to not to care about them. Frequently, they dry out completely in summer, so they are actually lakes. But they're actually hotspots of diversity, even here in Australia. So this diagram has one main goal, which is to show you the ecological functioning of these ecosystems, which are, to my view, hugely underrated, but crucial for even our economies and societies, and you'll see why I'm saying that in one slide. So very briefly, there's of course microbes in water, which play a key role in the nutrient cycling and energy flows within the ecosystem. And if we go upper in the food chain, there's macroinvertebrates. This is just a few of them: amphipods, annelids, copepods, crustaceans. There's one macroinvertebrate which is quite charismatic, and you might have heard of that, which is Artemia, the brine shrimp, which is actually one of the concos of why we can see pink lakes, for example, at Rottnest Island, or the famous pink lake down south, which unfortunately disappeared. Another key component of these ecosystems usually is microbial mats, which can be considered as megacities of microbes. And they have actually, from a biogeochemical perspective, a key role in terms of the carbon cycling, carbonates, deposition and release of oxygen. If we go upper in food webs, in the trophic chains, we can actually realize that this set of energy flows, food web interactions, and ecological patterns is shaping crucial conditions for the thrive, survival, and even breeding for wading birds. The most charismatic, for sure, is flamingos. The Atacama region is very rich in terms of flamingo diversity. We can find 3 different species. But we don't need to go further. We need to go to Chile to appreciate the importance of these lakes, because if we just think about Rottnest Island, the ecological conditions and the environmental setting that we can find through the hypersaline lakes that you might have crossed by bike, if you want to go to Little Salmon Bay, for example, they're actually providing a key component in terms of, again, surviving, thrive, and even breeding for wading birds coming all the way from Siberia. I'll go into that in a while. Now, even with this information, you might be thinking, yeah, why should I care about hypersaline systems, and why wetlands? Why? They're just a pond over there, and sooner or later they will disappear. Well, we all live in a climate change scenario, right? And decarbonization is a massive issue, and it is probably going to shape our near future lives. And if you think about wetlands overall, 30% of global soil carbon is stored in wetlands, so they're a huge carbon sink. Out of the total inland water volume that we can find on Earth, almost half of that is saline. So if we think about the effort that we need to invest in terms of fostering a sustainable planet, we're talking about a big component in terms of fighting or mitigating the effect of something that is absolutely impossible to avoid. Now, beyond these ecological characteristics and the importance from a purely environmental preservation perspective, hypersaline systems are actually quite interesting and important for recreational purposes, for example. This photo here is from the bottom left is from the Great Salt Lake in the US. Huge value in terms of recreational activities. Same if we think about ecotourism. In the Chilean Atacama region, for example, there's a huge, not huge, but important economic sector based on ecotourism, because these systems are quite unique in the world in terms of, if you just think about the colors of this photo, right? So there's tourists from all over the world wanting to go there. This is more or less the less impactful aspect in terms of economical activities on the lake. But of course, these lakes are hugely important also in terms of resource extractions. For example, there's the Artemia in B, top named B photo, the Artemia harvesting. Artemia is very, very important for aquariums. And there's actually a huge problem across the world in terms of spreading of just a few species. The pool of Artemias around the world is quite, it's not huge, but it's quite diverse. And there's a problem these days of people wanting to have an aquarium at home, getting bored of the aquarium and chucking the water of the aquarium in rivers and lakes. That has caused a dominance of just a few species of Artemia that are actually changing the dynamic of hypersaline lakes in a way that is detrimental for all the food chain characterizing these ecosystems. And of course, top left, resource extractions. Hypersaline lakes are getting more and more popular. mainly because of one reason. There's a huge demand for a green economy and a green future, and that comes with a cost for hypersaline systems. This is a research article. I was involved as an external commentator, let's say, on the potential impact of lithium extraction out of these hypersaline lakes on the populations of flamingos. And interestingly enough, they actually focused on 3 of the wetlands that we are targeting in our project. I'm talking about Salar de Tara, bottom right, Salar de Pusa, top right, and the Atacama, Salar Atacama, top left. This is actually a very interesting study, which I highly recommend, in terms of trying to find a way, a compromise in between resource extractions and also conservation of these ecosystems, because as we saw before, they play a crucial role in terms of decarbonization and storing the carbon that we are producing day by day through fossil fuels. Sadly enough, they found out that during the last 30 years, there's a decline in flamingo abundance across, especially across certain lakes, such as Salar de Tara and Salar de Atacama. Especially in the Salar de Atacama, there's the highest pressure in terms of resource extraction. The lithium industry is associated with a huge need for water. And if that is not done in a sustainable way from a flamingo perspective, but also objectively, that has an impact. So there's a huge need on conciliating this resource extraction need from a greener economy and also the preservation of these key species, which are, by the way, driving the ecotourism in the area. So there's a huge cascade effect in terms of detrimental impacts on the ecosystem, which are affecting local communities at the end of the day. A trend, more broadly, we focus on molecular tools in order to understand ecological dynamics. And my personal interest and our smaller group, let's say, is particularly focused on functional ecology. Functional ecology sounds great, everyone wants to do it, but it comes with a cost. The cost is that you need to be all the time up to date, and also you need to know a bit of a lot of things, which sometimes is a bit daunting, but it has a great potential and is quite rewarding. This is probably one of the reasons why, as you can see in this graph, the increase of functional ecology studies over the last, especially 10 years, has skyrocketed. And the other reason is that to date, especially starting from 5 years ago, we're starting gathering knowledge and expertise all around the world on different tools, molecular tools, biogeochemical tools, that are allowing us to unravel patterns that were impossible to detect or impossible to investigate before. One of them is certainly eDNA. The broader goal of this project is to combine that tool with other tools that I will explain in a second, in order to have the most complete idea on the ecological functioning of the systems that we want to study. Particularly, we are going to use eDNA, and specifically DNA metabarcoding, in order to unravel the molecular trends that I will explain in a while. But what's interesting here is that we want to combine that with stable isotope analysis. The goal of stable isotope analysis in this context is to focus on food web interactions. So we use stable isotopes as a fingerprint to understand the dietary preferences of flamingos, for example, and we want to see also the interactions in between, for example, microbes, macroinvertebrates, and all the way to the wading birds. A trend will be focusing on the molecular side of the project. Our collaborators at the University of Antofagasta, they actually have the only compound-specific stable isotope analysis machinery in all South America, and they will be carrying out conventional stable isotope analysis and also novel stable isotope analysis, which I won't go further in detail because it can be quite specific and not super relevant for this speech. We also collected, they also collected water samples, water parameters, nutrients, pH, conductivity, with the whole idea of combining these 3 branches, genetics, trophic ecology, and geochemistry, in a broader context and try to understand, as I said before, the functional ecology of these systems. Now, this is so far the theoretical side. The practical side came with a cost, as we all know. What we wanted to do was to collect samples from 3 salars, Salar de Pusa, Salar de Atacama, and Salar de Tara. They're all within the Los Flamencos Natural Reserve, which is actually perhaps the most important protected area in Chile. We're talking about 3 salars which are all Ramsar sites, and as we all know, Ramsar is this categorization which has international value in terms of preservation of wetlands. Why is that? Well, we focused on these 3 salars because they gave us, on the paper and also in practice, a nice altitudinal gradient that we wanted to test through the different tools that we are applying here. Salar Atacama is at 2,300 meters, and then we jump, in the case of Salar de Tara, to 4,300 meters above the sea level. So we are talking about high altitude lakes, especially if we think about Australia and how flat this continent is. And Salar de Pusa, just 200 meters above Salar de Tara, but in a completely different basin. The idea was to collect samples over 2 consecutive seasons. This was something that we brainstormed and we thought it was quite novel. There's virtually nothing done on these wetlands over consecutive seasons. But our friend arrived. And you might be thinking, here we go, another guy blaming on COVID. But seriously, it was really, really, really challenging. I won't go further in details. There's pretty crazy stories around this. We can actually tell you over beer. So that forced us to rely on just one field trip in 2021. Our collaborators in Antofagasta, they went in the field. I shared some photos a year ago, I remember, here. They went and collected some samples, both water and sediment samples. So in terms of numbers, just to give you an idea, we have samples from 3 salars, 27 sediment samples, and 49 water samples. A trend particularly, great, he's covering that side, but okay. So a trend particularly, we focus on molecular biology, molecular ecology. So we'll be using eDNA as a tool to unravel biodiversity patterns. And we want to screen these ecosystems through a series of assays that we know are targeting specific groups, or at least they're efficient with certain specific groups. So we'll use bacteria 16S for prokaryotes, CO1 for vertebrates and microorganisms and macroorganisms, but particularly we are interested in invertebrates, given the importance of artemia. And we do know that CO1 performs well with artemia. 18S, as Nicole said, a broad assay which is scanning the system, so we're curious to see what we get. 12SA for birds, and then we also want to have a look at algae. All this is work in progress. We received the samples just a few weeks ago as part of the kerfuffle created by COVID. So this project started as the other project almost 2 weeks ago, and it was like a diesel engine. It took a while to start. So as part of our friend COVID, as I said actually 1 year ago here in this room, it was actually a very nice experience in terms of challenging ourselves. And also an exercise of thinking out of the box. So we started brainstorming in the office, and we thought, okay, we cannot go there, samples cannot be collected, what can we do? We need to test this tool. Because, by the way, I haven't mentioned this before, but no one has ever done anything eDNA-based, purely eDNA-based from an environmental sample in hypersaline system, which is telling you 2 things. One, we are fantastic people. No. One, we are very good at targeting stuff that no one does. And 2, they're hugely underrated because we are talking about half of the volume of inland waters, sequestering 30% of the carbon in the soil that is probably saving us from a catastrophic near future. Anyway, a bit of positive note. I get frustrated because no one cares about the system. So we thought out of the box, and we went to Rottnest. Rottnest is a fantastic natural laboratory, 20 minutes away. You grab a ferry, you're there. A-class reserve, the highest level of protection in Western Australia. There's a side of the island which is more anthropogenic impacted, the touristic area, and then you have Salmon Bay, Little Salmon Bay, which is more pristine, let's say. So the idea was to test eDNA as a tool to investigate biodiversity patterns in hypersaline systems. And as I showed last year, we focused on microorganisms, and we have a study published as of May 2021. And over the last year, we focused on the upper levels in the trophic chain, particularly macroinvertebrates, and in the near future, we will be targeting migratory birds. The goal is to unravel the hypersaline ecology at the Rottnest hypersaline lakes and test some sort of functional ecology approaches that we will definitely apply also for the Atacama Desert. This is the latest outcome in terms of publishing out of this project, a paper led by the researcher and non-research technician Matt Campbell. And here we basically, long story short, we wanted to test environmental DNA tools and compare them with the classic conventional net surveying. So what we did, we went to Rottnest and we collected samples from the exact same 5 lakes that we targeted for microbial ecology analysis. And we collected sample with a net, which was the classic net that every consultancy person or regulator person knows, which is a mesh size 250, I think, if I recall it properly. And you just go through the water column and you try to collect and detect the most representative diversity of the systems. But what we also did, we collected water samples, which is the squared yellow icon over there, from the same spot. So with the goal to compare, hey, how effective is eDNA once we compare this what people have been doing for the last 150 years. We targeted the macroinvertebrate community, and we did it through the CO1 primer because we know it performs pretty well with Artemia and also with dipterans, which are more or less quite established, but they're very, very abundant and relatively diverse in hypersaline systems. So this is the first outcome of the study. Is a table, is a classic table that you can find in an eDNA versus something else paper, right? You have net and metabarcoding. What did you find with net and what did you find with metabarcoding? So the first interesting thing is that Artemia is pretty much everywhere and is detected both by net and eDNA metabarcoding. There's blooms of Artemia at Rottnest Island, and these Artemia, these macroinvertebrates, feed on the microbial mats and the algae, and the pink color that you can find, especially during the summer season, is partially derived also by the incredible abundance that you can find for Artemia at Rottnest Island. Now, stuff that we only found with nets. As you can see, there's a relatively high diversity, especially compared to the other taxa of dipterans. But there were Ceratopogonidae, Ephidridae, and also Stratiomidae, so 3 different families that were only detected through DNA through nets. But on the other side, we detected Haloniscus serlei, which is an interesting guy. Is originally from South Australia, actually, but is well established here in Western Australia, and is famous within the geek macroinvertebrate community for having a wide range of tolerance to salinity. There's also this guy, which is quite interesting. This is a beetle, more associated with coastal environments. Also, in terms of certain specific species of dipterans, Serpentine Lake, we could only find them through DNA metabarcoding. So there's multiple levels here of different tools detecting different things across the 5 lakes. There's, of course, stuff that we found in both, such as the Ephidridae family and the specific species that you can find here. But overall, this is telling us that something that is happening through the eDNA community and is more and more assumed and more accepted. eDNA is a tool that is complementary. it can be really, really, really powerful once you combine that with conventional approaches such as nets in wetlands or other conventional techniques. And this is more or less a snapshot of what we found. And the relevance of this for regulators or consultancies or anyone interested in studying hypersaline lakes is probably depicted by this graph. This happened at Rottnest Island, but it's actually the case of many hypersaline lakes across the world. You go there with a net, and visually, you pretty much can only find Artemia. The diversity spectrum is simply not there unless you're a specialist and you know how to collect specific specimens through different mesh sites nets. If you want to go up broad and have an overview from an ecological perspective on the diversity of these systems, the conventional stuff is always exposed to the risk of underestimating the diversity of the other taxa. And this is something that is, as I said, pretty common in hypersaline lakes. And the real value of eDNA within this context is to bring to light the diversity of the other guys that you cannot find, for example, Tanitasus in this case, or Haloniscus, as I said before. We went a bit further with Matt, Nicole, and the other collaborators in this study, and we wanted to check if the microbial patterns that we found at the hypersaline lakes and the differences in between these 5 lakes were actually matched by the eDNA data on macroinvertebrates. What we found, which is not super unexpected, we found out that there was a mismatch in between microbial and macrovertebrate patterns. This is not unusual because microbes are linked to the water chemistry and the energy flows and nutrients much more directly than macroinvertebrates. The species that you can find here, most of them have big ranges in terms of salinity tolerance. So the patterns that you can find in microbes and macroinvertebrates is a bit disconnected over there, but it's something that it wasn't hugely unexpected. Now, future plans. I have only 2 slides left. Hopefully there's plenty of time for a mind-blowing debate. Priority number 1, generate the data, which again, as I said before, it will be the first eDNA base generated for hypersaline lakes from these lakes, Salar de Tala, Salar de Pusa, and Salar de Atacama. Also, the samples are here with us, but we also have samples in Antofagasta, which will be targeted through isotopic analysis. I didn't go in detail during the speech on isotopic analysis. If anyone has any question, I'm more than happy to give more details about that. All in parallel to these 2 goals, there's a PhD student, as I said before. He is, at the moment, doing more conventional stuff. He's identifying the species under a stereo microscope, and he's doing multivariate analysis, crossing physicochemical parameters, nutrients in water, and checking if the different abundances are actually driven by certain specific environmental conditions across the lakes. What we are doing here, going back from Chile to trends, we have actually 2 different talks at the incoming in February eDNA conference in Hobart. The first one is this one, a title that we brainstormed with Matt, which is in between embarrassing and fabulous. Looking under the mat, benchmarking computational methods for analyzing microbial data sets. So to give you a bit of an overview, in this specific study, we focused on microbial mats, something that is probably the hidden gem of hypersaline systems because of their importance and because of their evolutionary and ecological role. Microbial mats are considered as natural, we consider them as natural laboratories for this study. Why is that? Because they're usually stable over time and they're well studied. So the idea we had with this study was to use them in order to have a biological community to test different molecular tools. We have present and past shotgun metagenomic data, which is probably the deepest scan that you can have from a biological community. And we wanted to test the validity of eDNA tools and compare the results that we get through metabarcoding approaches. And we want to do that by using two different platforms that you're probably not familiar with, PyCross-T2 and Tax4Fun. There's a lot of criticism in the literature because they're so-called predictive genomic tools. They're not assessing the functional patterns of expressed genes. They're just making a prediction on what potentially is the function that these microbes are actually developing. However, the beauty of these two tools is that you can create your own data set, and you don't need to rely on external data sets for environmental samples. So we'll be using metagenomic data to enrich the value of this functional prediction. And the overall goal is to compare outputs. And you might be wondering, yeah, beautiful, very technical, what's the point of this? Preliminary results that we already have are showing that by combining metagenomic data to enrich your dataset and use that customized dataset that can be applied through a metabarcoding approach, preliminary results are actually showing us that this could be a path, for example, for a lake that has been studied for decades. And having that metagenomic data as a starting point within a stable system could allow you to use eDNA tools to monitor that same lake over the years and create your own data set and assess the functionality of that specific community. This also is important in terms of the future challenges that microbial ecology is facing right now and will be facing in the near future. And Matt passed me the other day this literature review which was published this year, which kind of validated that what we are trying to do here has something in it. So in one of the last sentences of the paper, they state that the upcoming challenge, notably for environmental sample, will be to, don't remember? Yes, no, it's the same. Pretty much to link between functional prediction and reliable data sets. So at the moment, we do not have that. Perfect, thank you very much. To establish the link between functional prediction on reference data sets and environmental measurements. So thinking practically, government or consultancies, having that data set as a starting point on a relatively stable system like microbial math would allow you to collect samples and target just metabarcoding samples over there and create a functional investigation of the systems without having to rely all the time on the deepest technique that you have in the market right now, which is, by the way, the most expensive. Second, there's a second future plan, which is, the screen. Yeah, sorry. This is a second study, and here we'll be focusing on artemia. And you might think, oh, damn, again, artemia. I'm muted. Did I? What a mess. Okay, but this is not. It's not working, but yeah. Anyway, we'll be focusing on artemia. And there was a beautiful sentence with the mouse. Yes, thank you. Okay, so we're focusing on artemia. As I said before, artemia is very widespread, and there's this very, how do I define this? It's very grandiose sentence. Artemia cysts. Artemia during their life cycle, they have cysts. And actually, Matt the other day told me that there's this thing called about resurrection ecology. So these cysts last forever. So you can go there in the sediment and target ancient DNA and have a look and bring them to life from a molecular perspective. So these cysts are actually the most resistant of all animal life history stages to environmental stress, which I find quite interesting. And this is a sentence that I keep finding in artemia papers, which is probably one relevant guy mentioning it. And everyone, yes, you're right. But I believe that because they're amazing. They're amazing. And the goal of this specific study, and with this I finish, is to target Artemia because, as I said before, Rottnest Island is an A-class reserve. Why is that? Because we have quokkas, but also there's wading birds. These birds are migratory birds coming to Rottnest during the winter in the Northern Hemisphere. So they heavily rely on Rottnest, for example, amongst different environments in Western Australia and in Australia, for their breeding. They come and they need to eat on something. Artemia is the main dish on their menu. So they're the main food source for wading birds. So the goal is to check for the genetic diversity across these five different lakes, because we know that the microbial diversity is different. We know there's different patterns in terms of microinvertebrate dynamics. What's going on here? Are the birds preferring one specific lake, and what's their diet? We want to have very preliminary data that is showing that potentially the Artemia that we have at Rottnest is linked to the Artemia that you can find in the Northern Hemisphere. That is probably the best case scenario from our perspective as researchers, because that would indicate that is the evidence that there's flyway routes coming all the way from Tibet, coming here, that the birds are dispersing the Artemia, and is actually providing further evidence on the importance of Artemia for the diet and for the survival of these birds. We also want to target the gut content to see if they rely on different food sources across the different lakes, and overall with the goal of giving value to the systems, first of all, and to unravel certain dynamics that are hopefully allowing us to preserve these systems in a way that is compatible with, again, resource extraction, ecotourism in this context, and many other aspects that need to be considered within this very complex climate change scenario. With this, I conclude, and I thank you all for the patience.
Great, thank you very much, Mattia. Did anyone have any questions for Mattia while Euan gets mic'd up? All blown away. Has nothing online? Excellent. All right, well, if there's no questions, straight over to you, Euan. Bring us home.
How long have we got, Shane? 20 minutes. 20 minutes? Are we supposed to finish at 4? Yes. In that case, I'm going to switch from talking mode into a power lecturing mode, which is what I'm actually more used to doing anyway. So before we go any further, I want to acknowledge that a lot of Project 4 has actually been undertaken by Shirley Lucas, put your hand up or stand up, and Fred Wells. So we wouldn't have got anywhere without those guys. So we've also got Justin McDonald involved in the team. Justin unfortunately can't be here today, but the aims of the project are to develop reference sequences for key taxa for invasive marine species in Western Australia, and particularly focusing on the Northwest Shelf. Develop phyla-specific metabarcoding assays to better discriminate between IMS and native species. And as we've heard, the assays, well, actually it's not the assays, it's actually GeneBank that's actually a real problem. A lot of the sequences that are on GeneBank are either wrong or are missing. And if you want to find out more about that, have a talk to Fred afterwards, because he's actually one of the few taxonomists in the room who actually knows anything about marine invertebrates. And I guess the thing that we're really trying to do is to ensure that the assays can actually sensitively and confidently detect IMS in the marine environment. The cost of getting it wrong is a real problem, particularly if people are relying on it as a biomonitoring tool, and there are regulatory components around it. So this really basic and actually not particularly exciting work to many people is fundamental to anyone using eDNA in the marine environment. So the outcomes that we've done to date is Shirley has sequenced the mitochondrial genomes in the 16S region from identified pest species that the Department of Primary Industries and Regional Development have had. And we acknowledge Matt, who's sitting in the background, Claire, who can't be here, and others who have actually collected a lot of this material for us. And basically what we're also doing is collecting some of those native sister taxa that actually get confused, that we sometimes can't discriminate between. So by doing this, what we're trying to do is empower eDNA to actually be an IMS biomonitoring tool. And without it, there's a lot of hype, but not much substance. So we need to be able to confidently assign identify key pest species with a high amount of confidence. We need to be able to discriminate from the native species, which actually is really quite hard. It's easy to get to family, but sometimes it's really hard to get down to the species. And without that, eDNA as an IMS tool is not going to be that useful. We need to ensure that it's accurate across a range of different substates and also that it's cost effective. Otherwise, we're going to end up going back to more conventional techniques. So one of the goals is we actually want to make all of this information that we're generating with DPIRD publicly available, and Shirley's been loading it up to GeneBank. So that's underway. Also, one of the things that Tim was really keen on was to actually ensure that we had information and debate about eDNA with IMS and actually what it can and can't do that can actually be used to inform policy. So what have we done to date? Well, we've done a whole heap of things, but one of the things we have been doing is Fred and Shirley and another staff member have been up north doing collections. We know that from the Pilbara region alone, from what Fred's telling me, there's more than 5,000 species of invertebrates. Then we've got fish, and then we've got algae, and we've got other bits and pieces. Very few of these actually have genetic sequences. So Fred's been out, and we're lucky to have the skills of a taxonomist, collecting some of the sister taxa of things like oysters, barnacles, mussels, and some of the thayid snails. The 16S RNA region has actually been sequenced to full genome samples from both eDGES collections and from our own. We've been doing some of that, but we've also been using Craig Sherman's lab at Deakin University. And the data will be added to the reference bases as specimens are collected. We've still got some ongoing trips that we want to be doing. And Fred's just got back from Dampier on Sunday night from a particularly windy, rough, nasty trip. So we're using mitochondrial DNA gene regions because they have better detection rates for degraded environmental samples. Also, 16S tends to be well studied and well characterized, and there's more publicly available data for invertebrates. The other thing that's actually really important is it also tends to highlight more genetic variation, and this is really important for distinguishing between species. Shirley has done some work on testing some of the assays, and she's also tried multiplexing them. And I guess there's one key message that I've got here that'll keep it really quick. When we look at the multiplexing, so this is multiplexing for one assay that she's developed in singleplexing. This is for a second. We're actually detecting a lot of the, and you can't really see a lot of the key species which are in bold, but one of the things that we find with multiplexing, which we thought was going to be a way of reducing the cost and increasing the sensitivity, is that where we start getting species where we're getting relatively low reads, sometimes we're missing them. So last year where I stood here and said, multiplexing is going to be the way to go, I'm going to take that all back. I think we need to rethink some of that in the way that we actually do the analysis and really speed things up at a singleplex line. So that's one of the fundamental bits of research and something that Shane and I, a couple of others, have been working on over the last couple of months. We have tested these arrays with the DPIRD data. So DPIRD have a statewide array surveillance program which they deploy in a lot of the harbours by putting plates in. Those samples end up coming through Curtin. What we can do is we can actually run our assays over the material that's actually sampled from that. And this is another one of the surprising results. People often use the Leray CO1 assay, which is pretty good, or the 18S, and we've had multiple people say, yeah, look, 18S is really good at a broad level, but be careful. And we're delighted that some of the new assays that Shirley's been detecting are actually getting a lot. There are some things that they're not getting, but this is a really good starting point, and particularly given this, there'll be a lot more work on this. And taking this a little bit further, when we start looking at, I'll get that out of the way. 18S is pretty good at getting things at the family or broader level. Red is the CO1, can get some things down to a family level and species level. And the assay that Shirley's been developing is getting both things at a broad family level, but also getting down to species level. So we're pretty happy with that. It's very promising. And she said there's a big but. And that but is this yellow bit where there's a whole heap of stuff which is unassigned. In other words, there's a huge portion that we have no idea what's there. And that's because there's not the genetic sequences. And until people actually start focusing on getting those genetic sequences, it'll hold eDNA back. That's got to be one of the priorities. And then I think Fred, if he was here, would say, we need more taxonomists. We need more funding going into taxonomy. So I'll say that for you, Fred. It's been a common theme over and over again. So we've got papers coming out on oysters and barnacles. We've got taxonomy coming out on some of the particular species, and particularly the brachydontes. If you want to find out more about brachydontes, given the time constraints, talk to Fred. In summary, it's a mess. What people said was the right genetic sequence is wrong, and therefore eDNA can't really be used for brachydontes until these guys have redone the genetics, which will happen. Future work. Thank you, Mattia, for the COVID card. I'll play it one more time. We haven't been able to get overseas to do some of the collections that we wanted to do, so Fred and Shirley, or Fred and somebody, will be going to Singapore and Thailand, and we'll be bringing material back that we know are pest species for northwestern Australia, and then they'll be added into the assays. I'm going to move straight through. I'm going to do this one really, really quickly. Project 5 is actually using some of these assays and eDNA to actually look at the marine environment and do some sampling. Tim has asked us to focus on Port Hedland. And Port Hedland, because it's one of the busiest ports around, it's where a lot of BHP shipping comes in and out. And the aims are really, really simple, is to use eDNA to document the organisms associated with the different types of infrastructure, determine if there are differences in the compositions of the organisms, both on natural structures and across some of the, both on man-made structures and some of the natural structures, to test some of these new assays, and to compare and contrast the results with traditional 18S approaches. So, what do we know about Port Hedland and man-made structures? There are something like 7,400 man-made structures in Western Australian waters, and they are increasing and will continue to increase, particularly with renewable energy and other port developments. These are novel marine ecosystems, and they happen to be very attractive sites for invasive marine species, and particularly in the shallow water. There's not a lot of work that's been done on them. There have been a number of people like Chad Hewitt and some of the guys over on the East Coast that have done some work, but it's pretty limited. Port Hedland, busy place. It's got intense infrastructure in the port facility itself, and then you've got these lights which go out to, or channel markers which go out to about 20 nautical miles. The port structure itself, some of it's dredged down to around about 20 metres, so you've got a depth gradient. And we know with eDNA and just with marine invertebrates in general, there is a really tight depth gradient where things change even by over the scale of metres. That's got implications for the sampling we do. We've got to incorporate depth into the sampling. We also know that we've got spoil grounds. We've got a whole heap of anchor areas. We've got mangrove habitats, sand, and I know from doing some work up there with some of my colleagues at fisheries a little bit further to the north, areas that people thought were sand areas are actually sponge gardens. And these are some of the most rich areas around in terms of diversity, and probably exceed the diversity of some of the coral reefs. So we want to start targeting those areas. So we know we've got infrastructure, goes down to 19 metres. We know we've got at least 48 leading lights that go up to 20 nautical miles offshore. We've got 3 spoil grounds. We've got a whole heap of anchor areas. We've got sand, sponge, shoals and reefs that are definitely in that area, and a whole heap of mangroves. And we've got a certain budget that we're going to try and allocate that across. We know that we're going to have to use at least 2, preferably 3 sampling techniques. Water will give us 1 signal. And then we've got a couple of other techniques that we want to use, which will combine together into one sample. We know we want to use multiple assays. We're hoping to do a lot of the field work in July, August, September next year. But the first steps are to meet with Port Hedland and talk to the environmental officers, who happens to be one of my ex-students. Kurt, one of your mates, actually. Yep. And we've also need to talk to the harbour master about what we can and can't do before we get too far organised. So I think Fred and I are going to try and get up to Port Hedland before Christmas. Shane, I'm going to hand back to you, but before I do that, I want to acknowledge that we're going to have a few other people involved. Mike Bunce, who started Trend, Simon Jarman, who was one of the leads at Trend, Zoe Richards, who's from the museum, and Curtin, who's an invertebrate ecologist, Georgia Nestor, and then Tim's going to get involved in this as well because it's marine and he likes marine and he wants something. Is that right? Good. Is it you? Is it me?
Alrighty. Well, land speed record. That was impressive. Caught us right back up to speed.
Thanks, mate.
Thank you, Euan. All right, we'll get to interrogate him over beers in a little while. So was there any questions for Euan while we come to the end?
No.
No? Any online? No?
Perfect.
Excellent. All right, well done there. Okay, well, I don't have any substantial comments to finish. I don't want to stand between this and beer, so thank you very much for coming, and I would encourage you to stay around and to introduce yourself to many of you I know by name and I don't know you by face, so please come and introduce yourself and make sure you meet each other. But thank you to Tim and the BHP crowd for everything that you've done for the eDGES program. Still heaps more to go, we're only just warming up, so we'll look forward to inviting you back next year for the next round of updates. Thank you.